Good morning, students. Myself, Dr. Ragasudha, Assistant Professor and Head Department of Microbiology, Government City College, Hyderabad, is here with a new topic labeled antibody based techniques. Labeled antibody based techniques are also known as immunological assays. Immunological assays have always represented a major contribution in the diagnosis of infectious diseases. These techniques detect and quantify antigen and antibody in a given patient's sample. These techniques are applied in detection of various diseases like viral diseases, HIV, rotavirus, varicella zoster virus, Zika virus, etc. They are also used in detection of Lyme disease, syphilis, pernicious anemia, Rocky Mountain spotted fever, squamous cell carcinoma, toxoplasmosis, etc. Before getting into the details of the topic, let us go through the basic terms used in this particular topic: antigen, antibody, and antibody labeling. Antigen. It's defined as a macromolecular substance capable of inducing a specific immune response. The word antigen is originated from the notion that they can stimulate the immune system. While antibodies are glycoproteins formed by a plasma cell in response to antigenic stimulation and counteract with antigen with great specificity. It is the structure of antibody. Antibody is made up of 4 chains, 2 heavy chains and 2 light chains, and at the terminal end we can see antigen binding site. Antigen and antibody interact with each other, or when they come across with each other, they are specific, and when they are specific to each other, they form a complex known as antigen antibody complex, or this particular lattice is known as antigen antibody reaction. Next, what is antibody labeling? Antibodies are widely used to detect and quantify antigens in immunotechniques such as flow cytometry, ELISA, Western blotting, immunofluorescence, radioimmunoassay, etc. The antibody that recognizes the antigen is referred to as a primary antibody. While in these techniques we are going to use two types of antibodies, the antibody one which recognizes the antigen and the another antibody which recognizes this complex. So the antibody that recognizes the antigen is referred to as primary antibody. Primary antibodies are specific for different antigens. Antibodies used for detection of primary antibodies are known as secondary antibodies. The secondary antibodies are labeled by an enzyme conjugate which reacts with a substrate to produce a colored substance or a colored complex. The antibody bond covalently with the enzyme or the substrate. This is the image of labeled antibody. Antibody plus enzyme conjugate will produce a labeled antibody, and the substrate produces a color. The whole purpose of labeling is to provide measurability or detecting the concentration of the antibody or the antigen in the given unknown sample. Immunological techniques. Here there are two major types of immunological techniques to be discussed: ELISA, radioimmunoassay, and immunofluorescent assay. Today's class I will be discussing about ELISA. ELISA stands for enzyme linked immunosorbent assay. It was pioneered by two groups of scientists in 1972. The first group is Engvall and Perlman from Sweden. They have used this technique for quantification of antibodies. The second group is Van Weeman and Skurs from Holland. They have used the same technique for quantification of antigen. Later, it was developed by Clark and Adams in 1977. Salient features of ELISA. ELISA is a very sensitive immunochemical technique. ELISA is used to access the presence of specific protein which is in the form of antigen or antibody in the given sample. ELISA is used for quantification of substances like peptides, proteins, antibodies and hormones also. It is also known as solid phase enzyme immunoassay. It employs a enzyme linked antigen or antibody as a marker for the detection of the specific protein. Principle behind ELISA. ELISA is a plate based assay technique. The technique involves three principles which makes it one of the most specific and sensitive than other immunoassays to detect the biological molecule. The first principle is an immune reaction, that is antigen antibody reaction. Antibodies are very specific to the antigens and they interact with a specific antigen to form a complex, and this is the basic principle behind ELISA. The second principle is enzymatic chemical reaction, that is the enzyme catalyzes the formation of colored or chromogenic product from colorless substrate. Labeling is performed by organic dyes, fluorescent proteins, colored particles and enzymes like horseradish peroxidase, alkaline phosphatase, beta galactosidase, lactoperoxidase or tetramethylbenzidine. Enzyme substrates. Initially the substrate should be colorless. After degraded by the enzyme, it should be strongly colored or fluorescent. Enzymes like alkaline phosphatase, whose substrate is PNPP and the chromogen is PNPP, reacts with diethane diamine in presence of magnesium chloride, and this reaction can be stopped by 1 molar NaOH. The second type of enzyme is horseradish peroxidase. The substrate for this enzyme is hydrogen peroxide and the chromogen is tetramethylbenzidine plus phosphate citrate buffer, and the reaction is stopped by 1 molar sulfuric acid. The third enzyme is horseradish peroxidase. It is the most commonly used enzyme. The substrate is again the same, and here there is a different chromosome O phenylalanine diamine plus HCl, and this reaction can be stopped by 1 molar HCl. The reaction of a substrate in producing a colored substance. When tetramethylbenzidine reacts with hydrogen peroxide in presence of horseradish peroxidase, it produces an oxidized form of tetramethylbenzidine plus 2 molecules of hydrogen peroxide. Initially, before in a reduced form, tetramethylbenzidine is colorless. In oxidized form, it gets converted into blue color. This is a reaction being shown when on the membrane we can see the target protein on the membrane, and it is followed attached to a primary antibody. Primary antibody is in turn detected by a peroxidase labeled secondary antibody, and this peroxidase in presence of hydrogen peroxide, it gets activated and produces a color substance which can be detected. The third principle is signal detection and quantification. that is detection and measurement of color intensity of the colored products generated by the reaction between the enzyme and the substrate. The ELISA reader. The ELISA reader is the principle behind ELISA reader is almost similar to a colorimeter. Yeah, the source is received by a chromometer through a slit, and which is passed through a half mirror, which in turn is received by the cuvette, and later the transmitted light is detected by a detector. Now, let us move on to the procedure of ELISA. ELISA is performed in 4 basic steps. First step, coating; second, blocking; third, detection; fourth, reading results. Each step is followed by washing with phosphate buffered saline. Coating: a 96 weld polystyrene plate is coated either with antigen or antibody. An unrealitated protein is used to cover all unbound areas on the plate. The welds are added with test serum, positive and negative controls. Each conjugate is specifically targeted to antibody or antigen, followed by addition of a substrate. The signal or color produced by the enzyme substrate reaction is measured by ELISA reader or spectrophotometer at 450 nanometers. The color intensity gives an indication of the amount of antigen or antibody in the given sample. Picture of ELISA reader. This is another picture which shows the all basic 4 steps. First is coating. Polystyrene plate is treated with a solution of either antigen or antibody. Then removed liquid and plates were washed. Then blocking is done, and unrelated protein based solution is used to cover all unbound sites on the plate. Again the plates are washed. Then the third step is detection, where enzyme conjugate antibody or antigen binds specifically to the target antigen or antibody. Again the plates were washed and results were read. The substrate is added and the signals produced by the enzyme substrate reaction is measured with a ELISA reader. Once the substrate is added, we can see the change of the color from colorless to blue color. Here is another picture which shows the ELISA method. The plates were coated with different antigens, and the serum sample or the patient sample will be added, which consists of the primary antibodies specific for a particular antigen. They come and bind to the specific antigen, and once the primary antibodies were bound, secondary antibodies were added, which binds to the primary antibodies. These secondary antibodies, which are attached to an enzyme in presence of a substrate, produces a color. Types of ELISA. A number of variety of ELISA have been developed, allowing qualitative detection or quantitative measurement of either antigen or antibody. The technique is performed in 3 ways: indirect ELISA, sandwich ELISA and competitive ELISA. Indirect method. It detects the presence of antibody in the sample. The microtiter plate is coated with an antigen. It is a modified method of direct ELISA test. The primary antibody present in the sample binds specifically to the antigen after addition of the sample. The solution is then washed to remove unbound antibodies. Enzyme conjugate substrate antibodies were added to produce the color. The substrate is added to quantify the primary antibody through a color change. The concentration of the primary antibody present in the serum directly correlates with the intensity of the color. This is a diagrammatic representation of indirect method. The wells were coated with antigen, washed and add specific antibody to be measured. Again it is washed, then add enzyme conjugated secondary antibodies, wash the wells and add substrate and measure the color. Advantages of indirect method. A wide variety of labeled secondary antibodies are available commercially. Same secondary antibody can be used for detection of primary antibodies derived from single species. Disadvantages. Cross reactivity might occur with the secondary antibody, resulting in non-specific signal. An extra incubation step is required in the procedure. Sandwich method. The second method of ELISA is sandwich method. It is used to detect a specific sample antigen. In indirect method, we have detected antibodies. In sandwich method, it is used for detection or identification of antigens. So, the wells are coated with antibodies here. Non-specific binding sites are blocked using bovine serum albumin. The antigen containing sample is applied to the wells. Then they are washed, added with a specific primary antibody which sandwiches the antigen. So, hence it is known as a sandwich method, because the wells are plated with the antibody, then antigen is added, and in turn these antigens are bounded to a primary antibody. The enzyme linked secondary antibody is added that binds to the primary antibody. It is washed to remove unbound antibodies and enzyme conjugates. The substrate for enzyme is introduced to quantify the antigens. It is a picture of sandwich method. The wells are coated with antibody, washed, add antigen to be measured, again wash, add enzyme conjugated secondary antibodies and wash, and again add substrate and measure the color. High specificity, because the antigen is specifically captured and detected. It is suitable for complex or crude samples, as the antigen does not require purification prior to the measurement. It is a flexible and sensitive. Both the direct and indirect detection methods can be used. The next method is competitive method. This type of ELISA depends on the competitive reaction between the sample antigen and the antigen bound to the wells of the microtator plate with the primary antibody. First, the primary antibody is incubated with the sample. This results in the formation of an antigen antibody complex, which are then added to the wells that have been coated with the same antigen. After an incubation, unbound antibodies are washed off. More the antigen in the sample, more the primary antibodies will bind to the sample antigen. Therefore, there will be a small amount of primary antibody available to bind to the antigen coated on wells. The secondary antibody conjugates to an enzyme, conjugated to an enzyme is added, followed by a substrate to elicit a chromogenic signal. The concentration of the color is inversely proportionate to the amount of antigen present in the sample. This is a picture of competitive method. In this method, first the antigen and antibodies to be measured are incubated outside the well, then they are added to the well. The antigen antibody are added to mixture is added to antigen coated wells. So, only the free antibodies which unbound antibodies come and bind to the antigens which are coated onto the well. Then they are washed and a secondary antibody conjugated with an enzyme will be added, and again they are washed, substrate will be added to measure the color. Advantage: it is highly sensitive even when the specific detecting antibody is present in relatively small amounts. Let us see the comparison between indirect sandwich and competitive ELISA methods. In indirect ELISA method, the wells are coated with antigen. In sandwich method, the wells are coated with antibodies. In competitive ELISA method, the antigen and antibody which are to be measured are allowed to interact or react outside the well. In indirect method, after the coated wells, antigen coated wells are added with a sample of serum containing antibodies to be detected. Then they are washed and added with a secondary antibody enzyme conjugate, washed, and substrate is added to get the color. Whereas in and this indirect method is used to detect antibodies in HIV and HCV viruses or HIV and HCV patient samples. Sandwich ELISA. In sandwich ELISA method, the antibody coated wells were added with the serum sample with possible antigens, washed and added with a secondary antigen coated antigen enzyme, secondary antibody enzyme conjugates, washed and added with a substrate to produce color. And this sandwich method is used to detect antigens in tumor markers, hormones, etc. Competitive ELISA is used to detect antigen in free testosterones, that is, they used to detect hormones which are present in very less concentration. Here, the tubes, the wells which are coated with the antigen are added with antigen antibody mixture, washed and added with a enzyme conjugated secondary antibody. Again, they are washed and added with a substrate to measure the color. Applications of ELISA. ELISA based techniques are available and used clinically for testing for the early stages of cancer, including ovarian and breast cancer. The concentrations of illicit drugs such as cannabinoids, amphetamines, opiates, cocaines, benzodiazepines and methadones can be determined using ELISA in urine samples. The method is also used to monitor the levels of pharmaceutical drug concentrations in patients undergoing treatments. For example, anti-drug antibodies in rheumatoid arthritis and inflammatory bowel disease patients. It is used to test for the detection of hormone human chronic gonadotropin in urine, and it is which is usually present in higher concentration in pregnant women's. Collection of sample is very easy by this method. Detection of platelets antibodies in serum is used to identify patients suffering from disorders such as idiopathic thrombocytopenic purpura and systemic lupus erythematosus. These tests use the enzyme alkaline phosphatase, which is labeled with anti-human IgG. ELISA is also used to detect levels of potential food allergy contaminants at concentrations on the low parts per million scale. It also has the advantage of being able to test for oils and other substances like egg white or milk, which alternative methods such as PCR are unable to detect. ELISA was the first universal testing kit for HIV and employs the detection of human serum cystatin C to identify patients who are positive. The detection of West Nile virus is carried out by IgM antibody captured ELISA of patient serum on or cerebrospinal fluids, which has been taken 8 to 21 days following the appearance of symptoms. Thank you.